As we continue to assess the pandemic and what the state of the nation is, basically, we need to determine who or who has not been infected. To do these things, the antibody testing is required. So antibodies are our body's response, our immune system being activated, and in doing so is able to neutralize and stop the infection from taking place or clearing the infection. So companies are trying to develop a variety of different tests to begin to understand if your body is responding to the coronavirus. This has a lot of different questions about, am I immune? If I've had the infection, is this sort of acting like a vaccine? Am I not going to be able to get this again? A lot of these questions are a little bit unknown at this point in time. As the tests are being developed, they are looking again at these antibodies. So antibodies are extremely, extremely specific in how they interact with our virus, and it's not even the entire virus. They're acting with very small subunits of the virus. They are interacting with proteins, and even smaller level than that. So they interact with subsets of proteins or parts of the protein, which is known as an epitope. So a part of a protein is an epitope, and an antibody interacts with that. And so if you have a productive interaction from your body's perspective, you're doing something that's going to block that virus from being able to bind to your host cells or be able to replicate. And so having the ability to test whether or not your body has made these antibodies is important. And then further questions that come along with this are whether or not, if I do develop an antibody, how long lasting is this going to be? So that question is, do I have durable immunity? Right now, this pandemic has only been going on for 6 months, right? So it's really hard to tell if I have antibodies today, am I going to be immune from this infection in 2 years? So if somebody starts telling you information that they know on those fronts, I feel that that's a little bit presumptive on their part, because I don't think those questions are fully answered yet. In antibody testing, it is the goal to determine whether or not, again, whether or not your body has produced the correct antibody or antibodies to the COVID-19. So a test kit is going to provide a small piece of protein or an epitope for your antibody to interact with, and then it has to have a way to detect this. So a common thing that a lot of people are familiar with is a pregnancy test kit. So it's going to look something similar to that, except instead of using urine, you're going to use a drop of blood. And so the drop of blood is going to then diffuse across the membrane and have a chance to interact with whatever antigen is provided. And if it interacts, it's going to bind and stick there, and then you have a way to detect that. And then the antibodies flow further across the kit, and then if they bind, that's why you always have the control line. You always have 2 lines on those tests, whether it's positive, and then at the control, whether the kit is beginning to work at all. There are problems with this type of test, unfortunately. It is using a very small volume of serum, and so you're really unable to tell how strong of immune response you've developed. It's just whether it's there or not. So in that regards, that test is not very sensitive to the amount of immune response that you have. Other concerns with this type of test is that you can have a higher rate of false positive and false negatives. And these are really concerning because you don't want to think I'm safe from the virus or think I'm not safe from the virus. It just gives you bad information. And a lot of the information that we're seeing at this point is tests based upon this type of kit. And in doing so, we see numbers that are either over-reported or under-reported. It's really hard to have a true value or a true number of cases based on the test, I think, based upon the tests that are available at this point in time. As we move forward in the testing, there's also something known as an ELISA. And ELISA is a lot more quantitative than our lateral flow assays. A ELISA is going to take, again, a serum sample from your blood and interact with a specific antigen, just like the test strip. However, the main difference now is that we can be quantitative, we can do washing steps, we can do things that are a little bit more stringent, and in doing so you eliminate a lot of the false positives and the false negatives. And so if you have better data you have better answers. The other type of test that is available is a neutralization assay. And this would be where you would actually interact your serum with live virus and see if it is able to block it from infecting a cell in the lab. This is not going to be done on a wide scale level, most likely. Currently, any working with the virus in large quantities is of BSL-3, so it requires special gowning, special buildings, permitting, all sorts of stuff that make that test a whole lot more difficult to incorporate on a wide scale basis. So I think we're going to see the development of ELISAs. There are lots of companies, there are universities all over the country, probably somebody here even at SIU School of Medicine that is working on developing an ELISA test that will then be good to assess our patient populations. This is really important because if we know how many people are infected, then that can inform the decision makers on who is safe to go back to work. Are we developing to the point where herd immunity is being developed? And so herd immunity is where the entire population has enough antiviral resistance, or they're immune at a high enough level that just the amount of people that are immune is enough to restrict the virus from passing from person to person, because basically each immune person is a dot on our social distancing map. And so if I'm immune from the virus, then it's okay if I'm in between you and the next guy, because I'm not going to get sick. Unfortunately, those numbers are required to be 60 to 70% before herd immunity begins to kick in.